Intraneuronal energy deficiency, unifying mechanism in certain peripheral neuropathies. And the financial disclosure statement, I am a founder of Realm Labs. We make some products for peripheral neuropathy, some nutritional products. So the underlying theme of the lecture is going to be, is there an underlying mechanism common to sensory peripheral neuropathies of different etiologies? So you've got these different entities that come into our office, and they all have a very similar presentation. They have burning, tingling, numbness, shooting pains that progress in this dying back stocking glove distribution. It has this left-right symmetry. Is there an underlying process that would account for the fact that so many different diseases present this way? Now, there are differences in the presentation. Some come on very rapidly, some very slowly and insidiously. But what's going on? What's giving people these ulcers, this dysesthesia? And I'm going to speak to you about a mechanism that I heard about 3 years ago, and I've been fascinated with it. It turns out it works and coincides rather well with the work I've been doing, and it has to do with decreased energy availability into sensory neurons causing peripheral neuropathy. And the theory was first advanced by this gentleman, Gary Bennett, up in McGill, who worked on something called mitotoxicity, or damage to the mitochondria in sensory peripheral nerves. He's involved with chemotherapeutically induced neuropathy. And one of the thrusts of his work is that when people have chemotherapeutically induced neuropathy from the cancers, often that stops the treatment. The neuropathy gets so bad that they just stop using the agent. So it's very significant to find out what's going on, so you can give higher doses to save people's lives. And anybody who's really interested in this can read his paper. I'm going to be on the floor at the Tetra booth if you want to come by and speak to me if there's no time to speak here. So, sensory peripheral neuropathy. We're going to talk about the sensory peripheral neuron, which is the basic building block of the peripheral nervous system, a rather interesting structure. And it's very highly energetic. It needs a lot of energy. Neurons are very hungry for energy. The analogy is that the brain is, the neuron is in the brain, 2% of your body mass, and it's 20% of your blood. So it's well known that nerves take a lot of energy to work, and that's because they're involved in conducting an action potential. And what's going on there? This is a rudimentary diagram. You've got the exterior and the interior of the nerve cell with the neuronal cell membrane in between, and you've got this ionic gradient between the potassium and the sodium, which causes the action potential to be conducted along the membrane as the sodium ions flow in, and you got this depolarization and repolarization. And in the afferent neuron, that goes to the brain, and the brain interprets that and figures out what's going on in the extremities. Well, paradoxically, what I just showed you takes no energy at all. The sodium channels open up as a result of the electronic, the voltage changes, and sodium just flows in. What does happen, though, is this has to be cleaned up. You have to go ahead and reestablish this ionic gradient. And the way the body does that is with this mechanism, with a sodium-potassium pump. A sodium-potassium pump is this cell-resident structure, a macromolecule that takes a lot of energy, and it's squeezing out the sodium and pulling in the potassium to reestablish this ionic gradient so that the next action potential can be sent. And if you think about this situation, this structure here, the longer the neuron, the more of these macromolecules you need, because this is a very localized event. You're cleaning out the sodium ions within that spot within the neuron. So the longer neurons require more of these little macromolecules, and the shorter neurons require less. So the longer the neuron is, the more sodium-potassium pump energy is necessary. And it turns out that takes the majority of the energy necessary to run the cell. Over 60% of the energy needed to run a neuron is devoted to the sodium-potassium pump, probably even longer. And that's related to the size of the neuron. The longer neurons, just like a copper wire, the longer the copper wire, the greater the resistance. It's a great analogy, I just heard it today. Requires more energy to run it. And this stuff was worked out long ago, and it's been refined. This work is 50 or 60 years old. And I want you to think about the the neuron that's running to the tip of my big toe. It starts in my spinal cord, it runs through the dorsal root ganglion where the cell body is, and goes to the tip of my big toe. Now I'm 6 feet tall. And the most common neuron in the peripheral nervous system is the C fiber. The cross-sectional diameter of a C fiber is a micron, a millionth of a meter. So you've got an anatomy here which is really rather remarkable that we treat. This is a structure that's 1,000,000 by 1. The length of that would be analogous to 100 meters of 5/0 nylon. Think about how fragile that structure is, how amazing it is that that can conduct this action potential from that massive distance, those proportions. And that's what we're dealing with. And where is this structure getting its energy? Well, the vast majority is coming from a mitochondria, my favorite organelle. And the mitochondria, you have glucose coming into the cell. This is the way we produce energy. In the cytoplasm, there's some glycolysis, you get some ATP, but the results of glycolysis, the end products, go into the mitochondria and produce this explosion of ATP. And that's what makes us run. That's what makes us multicellular, that's what makes us differentiate, that's what allows us to be the organisms we are. This is basic to all eukaryotic cells, this sort of structure, except for maybe red blood cells which don't have mitochondria. So Bennett, in his work, and he's the pioneer here, he first worked this out in chemotherapeutically induced sensory neuropathy. And I was fortunate enough to see him 3 years ago when he lectured before he published. And it was very influential for me. So he talks about chemotherapeutically sensory neuropathy. And this is the study he did way back when. And you're welcome to any of these studies, just come by and see me. And it turns out that what I'm going to be talking about, the process I'm going to talk about, is not just unique to one element. There are 4 different types of agents, 4 groups of agents, that cause chemotherapeutically induced peripheral neuropathy. And the characteristics, as we all know, tend to be what we see. This bilateral symmetrical neuropathy, it sends up the feet and legs in a stocking glove distribution, and it's correlated to how toxic the agents that you give somebody is. If you give them really a lot of very toxic agents right away, you get a lot of peripheral neuropathy very rapidly. If it's more slowly, the progression of the neuropathy occurs more slowly. And what's happening there? Well, this is a great slide that Bennett shows, and this is a nerve fiber, a sensory nerve fiber. I think it's a sterile nerve fiber in an animal that was given paclitaxel. And if you'll notice, those fibers and those big oval things with the arrows pointed at them, they're mitochondria. So the mitochondria in yellow look completely normal, but the ones that are in red are vacuolated. They're diseased. They don't have the ability to make ATP. And it's his thought that as a result of these mitochondria not making ATP, you have significant energy depletion in that nerve, and that's what causes the sensory neuropathy. And the yellow ones are well, and the ones that are in red are the ones that are vacuolated. And this is the study that just came out. I recommend you read it. So he developed this theory of mitotoxicity, which is a chemotherapeutic insult to the axonal mitochondria, reduces ATP levels. The question becomes, what are the consequences of reduced ATP levels in the sensory nerve? Well, the energy hog in this system is the sodium-potassium pump. We just went through that. So you're not going to be able to maintain that ionic balance across the cell membrane as successfully as you had. And what's going to happen? Well, you're going to have spontaneous action potentials created. And if it happens to be in a C fiber, your brain's going to interpret that as pain. And if it gets really bad and you're unable to conduct any action potential at all, you're going to go numb, which is exactly what we see. You're also going to have an inability to support the structures of the intraepidermal nerves because they're very energy dependent also. The reason is that these interdigitated nerves, this end arbor on the neuron, goes through this stratified squamous epithelium, and as that's reduced and it's sloughed off, the nerve fibers have to regenerate. It takes a lot of energy, a lot of mitochondria at the ends of the nerves, and you can't repair them as well. And we see this clinically all the time as decreased epidermal nerve fiber density. And this is a 3-dimensional thing. We look at it as planar, as 2-dimensional, but it's these nerves come up in this arbor, and they're constantly being damaged by the squamous epithelium as it's going up. This takes a lot of repair. And also you'll have decreased ability to transport stuff from the cell body down to the tip of the axon, because the vital cargo that's essential for the nerve to work properly is produced up in the cell body. And then you have this system, which is axonal plasmic transport, where this stuff is taken from the cell body and transported down, including mitochondria, we believe. They're produced higher up and transported down. And it's done by this rather amazing system, which was discovered maybe about 25 years ago, these motor proteins that physically walk down these tubules, and they carry these liposomes or these mitochondria, and that breaks down as well. So that's what happens in chemotherapeutic-induced neuropathy. Let's talk about HIV-induced neuropathy. Well, this happens, the characteristics of an HIV neuropathy are very, very similar as well. And it seems to be less rapid onset than chemotherapeutically. And the reason that is because there are two reasons you'd get HIV-induced neuropathy. One is the proteins of the virus itself are mitotoxic. They actually damage the mitochondria. And secondly, the agents that we use to treat HIV disease are also mitotoxic. So the more agents you give and the more of the viral load you have, the more rapidly this will progress, and it's what we see. And this study was done out of Texas. It talked about mutations in mitochondria, and the progressive number of mutations, the more distal you got. So there's good evidence that mitochondria are significantly damaged in HIV disease. And that mitochondria damage decreases energy availability to those cells, and it expresses itself clinically as a bilateral symmetrical peripheral neuropathy that ascends the way we see it. What about diabetes? Well, there's plenty of evidence about diabetes. Diabetes has also a bilateral symmetrical neuropathy. It ascends up the feet and legs in a stocking glove distribution, and its progression is much, much slower. Why? Well, because the things that cause complications in diabetes, which has been worked out to be basically the activation of these pathways, well, that sort of disease and damage that it causes occurs more slowly. It's a much more complex disease. Excuse me. The main factor, the main molecule that causes the activation of these pathways, which are the complication-causing pathways, has been worked out to be the superoxide radical. This is formed inside the mitochondria. This thing is formed in everybody. It ages you naturally, but when you have diabetes and excess sugar flowing through the mitochondria, it works it up much more rapidly, and this bounces inside the mitochondria, causing damage physically to the structures in the mitochondria. So this is the prime mover here, and it's made in the mitochondria, and it's damaging the mitochondria. That's why the progression is much more slow. At least that's the theory. And there are a lot of papers on mitochondrial disease and sensory dysfunction in diabetes and sensory neuropathy. This is not new stuff. This is just a correlation of stuff that's been out there. And all these papers are available to you should you be interested. And Bennett talks about diabetes, and it goes through the case, and he says the data suggests that in diabetes, sensory neurons are unable to produce enough energy to meet the demand of bringing that action potential and those reasons why you need a lot of energy to support the function of the cell. And you also, if you think about the length of the cell, the length pattern, the longer the cell, the more energy you need. So if you have a situation where you have less energy available than you should, that system's going to fail by those parts, sequentially by those parts that need more and more energy. So the longest neurons are going to fail, and then the ones that are a little bit shorter are going to fail. The ones that are a little bit shorter are going to fail in an environment where there's less energy than is needed to make that system work. And that's exactly what we see. So there's idiopathic neuropathy, which we all know about, and I think that's a very poor term because we include very often people who are old and whose nervous system are breaking down, just aging, and since we don't have a diagnosis for them, we say they're idiopathic. And they shouldn't be idiopathic. I think we should reserve that term for people who have a paraneoplastic syndrome they haven't figured out the cancer yet. They walk into your office, they have a brutal neuropathy. We don't know what they have. Let's call it idiopathic, which means we don't know what they have. Or somebody's got sarcoidosis that isn't present clinically, or amyloidosis that isn't present clinically. These people have idiopathic neuropathy. The vast majority of people we call idiopathic are old. They walk in, I got some tingling, got some shooting pains, I got some numbness, it's been getting worse, it started in my big toe and it's progressing over the last few months, years, etc. These people have senile-induced neuropathy. As you get older, your nervous system breaks down like every other part of your body. When it happens peripherally, it presents as a peripheral neuropathy. When it happens centrally, you get dementia. So, and it's very interesting that as you get older, the percentage of people with neuropathy progresses dramatically. From 65 to 74, it's about a quarter of us. And if you're over 85, your chances are better than 50% that you have a neuropathy. Now, this neuropathy, which progresses, also progresses in the same sort of pattern. This stocking glove slowly ascending neuropathy. So you would expect that there would be some mitochondria or energy issues involved with getting older. And that's exactly right. It's very well documented. And these papers are available. I can't read, though, mitochondrial dysfunction in elderly and possibly with respect to insulin resistance. The paper shows that on older people, when compared to younger people, there's 40% less mitochondrial activity or ATP production. It's another study, this is out of Denmark, I believe, which showed that falling ATP levels in mitochondria contributes to the aging process. But you really don't need to be a neuroscientist to know that as you get older, you get weaker. You don't have as much energy as you used to have when you get older. It's obvious. And the way that impacts the nerve is by a peripheral neuropathy, which ascends the same way your energy depletion progresses. And finally, thiamine deficiency-induced peripheral neuropathy. I think this is an epidemic that's completely undiagnosed, that is unrecognized, and we're making lots of people better by giving them thiamine derivatives. And I think the reason is that they tend to be thiamine deficient, and we fix them and it works rapidly. Now, if you have somebody who has thiamine deficiency-induced peripheral neuropathy, their symptoms are similar. Bilateral symmetrical, ascends from the toes up the feet and legs, in a stocking and glove distribution, with pain, burning, tingling, etc. If that person became thiamine deficient because you put them in a Japanese prison camp during World War II, they get beriberi like that, and they get really brutally painful. If they have a disease process where they're urinating out their thiamine, they get the symptoms much more slowly, and that's what we're seeing. And there's evidence that thiamine deficiency in and of itself damages mitochondria, but I think more important here is the fact that thiamine is absolutely essential for the mitochondria to work. It's a mitochondrial fuel. 90% of the thiamine in the body is in the mitochondria. It's absolutely essential for 4 of the prime enzymes that take energy, extract energy from sugars to work. And if you have decreased thiamine levels, you have decreased ATP. Remember, it's hoarded in the mitochondria. 90% of it is in the mitochondria, and that's because the enzymes that use it are in the mitochondria, producing ATP. So in the example before about glucose coming into the cell, going through glycolysis, you get a couple of ATPs, it goes into the mitochondria, you get this explosion of ATP. If you don't have thiamine, you don't get that explosion of ATP. So you could think of, if you wanted to stretch the analogy, as a chemical mitotoxicity. But in any case, you have decreased energy availability to the cell, and that's causing it to break down in the sequential manner that we spoke about. And in addition, when you have thiamine or B1 deficiency, you have 2 categories that we see all the time that are known very well to be endemically thiamine deficient, and also have a high percentage of them have neuropathy, which is the elderly. They have 75% less thiamine running around in their plasma. That's well documented. And thiamine deficiency in the elderly, along many other vitamin situations, is also well documented. So, you've got this complex, this sensory peripheral neuropathy that's caused by many different diseases. The 5 diseases that I mentioned to you is probably 90-95% of the neuropathy you see in your office. And it's always a bilateral symmetrical progression, or often. There are different variants. Some people get chemotherapeutically induced neuropathy, they'll get pain in their cheek, or they'll get another peripheral nerve that breaks down. But in general, it's a rather common progression. And it's associated with the nerves that need a lot of energy, these peripheral nerves, and it tends to break down in the same sort of pattern as the pattern that is necessary for the nerves to work properly. The longest nerves breaking down first because they need the most energy, and they're progressing up, and it also accounts for the symmetry of this because your size of your nerves are the same on each size. So what we're talking about is this overriding unified mechanism. I know nobody came here for electron bioenergetics, so what do you do about this? How do you take this back to the office? Well, I think we as a profession, generally we suppress nervous system function when we have to. We give anti-epileptics, and we give antidepressants, and we give opioids, etc. But I think when you think about this sort of theory and this general model, it makes a lot more sense to try and supply the mitochondria with what they need, whatever nutrients are necessary to make them as efficient as possible. I think you can take it home on Monday. Thank you very much.